Okay, let's study different types of sclerenchyma now. So the sclerenchyma is basically divided into two types on the basis of form, structure, origin and development. On the basis of form, origin, structure and development, the sclerenchyma is divided into two types. They are sclerids and fibres. They are sclerids and fibres. Okay. Right. Let's study the differences between sclerids and fibres. Okay. So sclerids are, the shape of these sclerids is generally they are cylindrical. They are cylindrical or oval, or they can be polygonal. They can be cylindrical, oval, polygonal or spherical. So these are different shapes of sclerids. Whereas fibers are elongated cells with tapering ends. Elongated cells with tapering ends, having tapering ends. Okay, now let's see the structure of this sclerid. One sclerid. So this is the sclerid, and the sclerid consists of, sclerids are with highly thickened cell walls. Sclerids are with highly thickened cell walls. Due to the presence of this highly thickened cell wall, the lumen becomes very narrow. So the lumen of the sclerid becomes narrow due to the presence of highly thickened cell walls. Right. So this is the lumen, and this is highly thickened cell wall. Highly thickened cell wall. Okay. So at some points these thickenings are absent in this cell wall. At some points these thickenings are absent. The points where these thickenings are absent in the cell wall are called as, these areas are called as pits. These areas are called pits. Pits are unthickened areas of the highly thickened cell wall. These pits help in transport of water. So these are pits, unthickened areas. Now let's see the structure of fiber. So even fiber is also a highly thickened, also a cell which is elongated cell with highly thickened cell wall. Like this, highly thickened cell wall. So here also cell wall is highly thickened, but the thickenings of the walls of these fibers are lesser when compared with the wall thickenings of this sclerids. So these thickenings are made of lignin. These thickenings are made of lignin, and here also these cell walls are with some unthickened areas. So here also pits are present, unthickened areas are present, which are called pits. So this is one fiber, right? So this is pit. This is lumen, and this is highly thickened cell wall, which is made of lignin. Highly thickened cell wall made of lignin. Okay. Now what is the function of this sclerids? Function let's study at last. Now these sclerids can be branched or unbranched. Sclerids can be either branched or unbranched. They can be branched or unbranched, whereas fibers are always unbranched. Fibers are always unbranched. They are not branched. Next, coming to whether they are present singly or in groups. Sclerids can be present singly or they may be present in groups also. So they may be present either singly or in groups. Next, Coming to fibers. These fibers are always present in groups. These are always present in groups. Okay, next coming to functions of these sclerids and fibers. What is the function? Sclerids provide stiffness to the plant organs. They provide stiffness to the plant organs, whereas fibers provide mechanical strength, mechanical support to the plant organs. Mechanical support to the plant organs. Let's see where these sclerids and fibers are present. So sclerids are present in the pulp of some fruits like guava, sapota, pear, and they are also present in the seed coats of legumes, and the sclerids are also present in tea leaves. They are also present in tea leaves, and they are present in the walls of nuts, dry nuts. So these are the locations of sclerids. And as these sclerids are present in the pulp of some fruits like guava, pear, sapota, when we eat those fruits, we feel gritty. This grittiness is because of the presence of sclerids, which are providing stiffness to the fruit, pulp of the fruit. And fibers. Fibers are present in xylem and phloem. Xylem fibers and phloem fibers, which are involved in providing mechanical support, mechanical strength to the plant parts. So this is about sclerids and fibers. Now let's study some more different types of sclerenchyma. Sclerenchyma. Okay, now let's study different types of sclerids. So sclerids are of 5 different types on the basis of their shape. Brachysclerids. These brachysclerids are isodiametric in shape. That means they are similar at all sides. So they are isodiametric in shape and they are present in pith, cortex, bark and even in the phloem. And they are also present in pulp of some fruits like guava, pear and sapota. Due to the presence of this sclerids in the pulp of these fruits, they give grittiness. We feel grittiness. So they give stiffness to the pulp of the fruits. As they are giving grittiness, these are also called as stone cells. Brachysclerids are also called as stone cells. Now next category of sclerids. Next type is macrosclerids. These are also called as rod cells because they are rod shaped sclerids. And these type of sclerids are present in the seed coats of legumes. Next type, third type, that is osteosclerids. These are also called as prop cells because their shape is barrel shaped. They are barrel shaped and hence these osteosclerids are also called as prop cells. And these are present in the seed coats of monocots and they are also present in leaves. Next, coming to fourth type of sclerids, they are asterosclerids. These are also called as star cells because they are star shaped. They are in star shape. Okay, these are present in the petioles of hydrophytes like nymphia, lotus, and they are also present in the leaves of thea, that is tea leaves. Okay, next coming to last category of these sclerids, they are trichosclerids. These are hair-like. They are long, branched and hair-like sclerids, and these trichosclerids, and hence actually trichomes are long. They look like hairs. So trichosclerids are long and they are branched and they are hair-like sclerids, and hence they are called trichosclerids. These are present in hydrophytes and aerial roots of a plant called monstera. So this is all about different types of sclerids. So till now we studied about different types of simple permanent tissues. They are parenchyma, collenchyma and sclerenchyma. I explained these 3 types of simple permanent tissues by making differentiation, that is comparative study. Then we studied about different types of parenchyma, different types of collenchyma, and then we studied about different types of sclerenchyma, that is sclerids and fibers. Now we studied about different types of sclerids also. So with this, different types of simple permanent tissues is completed, and next I will be starting with complex permanent tissues, that is xylem and phloem. Hope you understood this concept. Thank you, students.